Thanks very much, Beth. And hi everyone again. Um, so I'm kind of quite excited to tell you about the accelerator. So it's been a program that we've been working up now for probably 2 years, and was funded by Cancer Research UK last year. And we kind of were about to, well, we did launch in March, but then the world changed and it seems like everything has kind of slowed down a little bit, but we're working away in the background to get things off the ground. So I'm going to give a little bit of an introduction. You guys don't really need this background, but I think the points are worth making that, meso in the UK is the lesser known epidemic, if you like. And for decades, it has been underfunded. We've really suffered relative to our other well-deserving cancers in terms of pounds spent on lives lost. And Dean's talk is kind of inspiring because we start to see things actually changing. We start to see maybe 1 year survival starting to shift, but it remains a cancer of unmet need, and there is a kind of global epidemic on the horizon, because the images you see there are young children and adults working in the BRIC countries, so that's Brazil, Russia, India, China, where there is really no asbestos control, and so there is a kind of an epidemic looming. The Dean's kind of talked us through the kind of exciting developments over the last 12 months, but really, this is a review article published a few years ago that just looked at the numbers of trials active and what phase they were at, and there's a positive trend there, which is a good thing. You've got more trials running, but you can see in terms of where those trials are that very few studies or very few research questions or drugs get to phase 3, and phase 3 is of course where we test whether a drug actually works better than what we currently do. And there are lots of reasons for that. But one of the reasons, I think we would all agree, is that we have not, over the last 20 years, understood the disease in a way that's allowed us to really design therapies in the best possible way. And therefore, we're more likely to fail in patients. It's much better for us to fail in the lab before we go into a patient. Our understanding of the disease has improved incredibly, and you'll recognise some of the broad groups of drugs here. Immune checkpoint blockers and arginine deprivation were picked up by Dean. Viral therapy is probably not going to excite anyone at the moment, but oncolytic viruses are another strategy that we've tested, and anti-angiogenesis drugs have certainly been a kind of significant flavour over the last few years, and antigen-based vaccines also. The difficulty has been translating our knowledge from the laboratory, or our knowledge about a mesothelioma tumour cell, into designing an effective drug. And so for those of you who are interested, that there are these kind of important publications that have really allowed us to really describe the mesothelioma genome quite comprehensively now. And the figure that's plotted there in simple terms is the number of mutations per cancer cell by different cancers. And mesothelioma is the third one along on the left. So it's a cancer that doesn't actually have, relatively speaking, as many mutations as others. And in many ways, that's a good thing. And we would think that we could, if we could understand that mutation, we could drug that mutation and we could cure the patient. The real challenge in meso is that the mutations that occur are not mutations in oncogenes, and those are genes that drive a cancer's growth and life cycle. They are quite commonly mutations in what are called tumour suppressors. And tumour suppressors are a bit like the brake of your car. Not having a brake doesn't necessarily mean your car's going to accelerate. And so it's been difficult to understand how just not having the brake applied, not having a functional tumour suppressor, has translated into a cancer that kills patients so quickly. And it's therefore been quite difficult to design therapies. And we've extrapolated a lot from other cancers rather than had bespoke programs like Dean's. And the genome is also dominated by a very obvious inflammatory signature. Inflammation in the immune system seems important. So we really designed Predict Meso really from that position. And, you know, this scheme at the top that you'll recognise, it's a little small on my screen, but you might, I suspect you can get the idea, where we have usually 30, 40, thank you, 50 years of inflammation. And then at some point, we have the evolution of mesothelioma. And it's not clear what happens there. There is clearly a final critical last mile where maybe the immune system fails to stop killing those little tiny mutations that we all develop in our life, or some other inflammatory process develops, or something happens. And understanding that last mile gives us an opportunity to drug patients more effectively, but also perhaps to intervene earlier. And in the second little figure there, you've got a description of something called benign asbestos pleural effusion. So that is a phenomenon that the chest physicians here will recognise, where patients present with fluid and it looks like it could be meso. We biopsy them, but it's not meso, it's just inflammatory change or fibrosis. But we all follow those patients up because we know at some point they may develop meso. And there are some subtleties to that as to whether that was really a meso all along, but the reality is that that window of opportunity is that one that we can exploit, because we often have inflammatory biopsies in these patients just before they develop a meso. And that paired material is potentially incredibly powerful for us understanding what changes. And the other really exciting thing that's happened in the last 5 years, 3-4 years, is Marion McFarlane's lab in Leicester have a mouse model, which is kind of described by the little cartoon at the bottom. And in their mesothelioma mouse, if they inject asbestos into the pleural space, they can watch over time and they can actually demonstrate that some of the genetic features that we see in the clinic in established mesothelioma tumours are actually present in the pleural space before the cancer invades, or even before it becomes cancer. And so there is potentially this opportunity to intervene early, because some of these events are maybe obvious in patients before we see them. So the accelerator was funded last year and provides us with £5 million over 5 years from predominantly CRUK, but also CRUK's compatriots in Spain and Italy. And the aim and questions are reasonably simple, in that our primary aim is actually to build these critical tissue cohorts of benign cases matched to mesotumor, and also to build technologies and infrastructure. And the infrastructure particularly includes really robust, well-designed preclinical models, so that we at the end of 5 years can get to a point where we have more drugs ready to go into a mesothelioma trial network, a bit like Dean's MIST platform. And we would really see these things as being running in series and together. And the questions that underpin that really are: how does inflammation evolve into meso, and what are the key molecular events? And importantly, what are the vulnerabilities associated with those events? Dean gave the really great example of arginine deprivation, which is a vulnerability in ASS1 mutated mesocells. And those are the sorts of things that we would be looking to see. And we also want to try and understand who might the best patients be to recruit to even chemoprophylaxis studies of high-risk asbestos-exposed patients. And so we need to understand risk, and we need to understand response, and I'll kind of talk you through So this is our map essentially of the network, and given the nature of the funders, we are predominantly based in the UK, Italy, and Spain, although we have collaborators in Belgium, and we have a significant collaboration in Brazil in imaging, and that's important because Brazil, of course, are one of the big countries that are going to be faced with meso in the next few decades. And we've already had contact from groups in the US and other countries, and it is a network that we hope to grow and expand and share. And if you just look at the image in the center there, the network is a network in the sense that we aim to share everything, and that includes data and tissues and models and information. And it's also a network of work packages which are linked together. And so we have 5 work packages. And so in the time I've got, I'm just going to show you a single slide, giving you a kind of brief overview of what each work package is going to try and do. So the first work package is the really critical step in actually building these cohorts of benign meso matched tissue pairs, and using that tissue to understand downstream in the other packages how that meso has evolved, and also to build models using that tissue. There's a really important data sharing piece. Crispin Miller in Glasgow, who's our bioinformatics lead, is essentially building a sharing infrastructure, and we'll use ICGC Argo to share a lot of the sequencing and other data out with the network. We've placed a lot of importance on developing other new researchers. So we have a series of schools and seminars planned, and we have 5 PhD students who are spread across the countries, including 2 that are coming from Brazil. And the reason for that is it's for the sustainability of this, and for this to grow, it's the people that will make it grow, much like our clinical network in Scotland. And so us developing these people, I think, will help us move the thing beyond the life of the actual accelerator award. The schematic there is just illustrates that the tissue collection effort is necessarily complex, in that we are collecting prospectively, and we'll do a large prospective study in the UK involving 25 centres, where we recruit patients with benign pleurisy and follow them up. And we also will have a retrospective element, which is going to be really housed within Mesobank, which is Robert Rintoul's labour of love. And Robert is going to build those tissue pairs for us in Cambridge. So the second work package is really our omics. And so the intent here is to use some really incredible researchers across Europe: Andrew Bianchen in Glasgow, Susanna Cedrez in Barcelona, Manela Stellar also in Barcelona, and Seth Kofelt in Glasgow, to help us understand what processes are allowing or driving the evolution of meso. And so those broad groups are the genome, so those are really the development of mutations in the DNA, the epigenome, which is more to do with the switching on and off of genes, and the transcriptome, which describes what proteins are expressed and whether those are actually active in the cell, and then the immunome, which is really describing the immune landscape around a mesothelioma tumour. And that's really critical in meso, because our pathologists constantly tell us that most of the biopsies that we send them are stroma, they are microenvironment. It's inflammatory cells, fibroblasts, and the odd tumour cell. And so we hope to try and understand that over the 5 years. So work packages 3 and 4 are together on this slide, and this is a hugely shameful simplification, but essentially the material that we generate from the patients will be used to generate preclinical models. And preclinical models essentially have various forms, but they can be as simple is the wrong word, but they can be as clean or as pure as a patient-derived cell line where it's just tumour cells. That's harder than it sounds. Or it can be an organoid or an organotypic model where we try and have a three-dimensional model that has tumour cells but also has some of these important immune cells. And then we have more integrated models that might be within a mouse, and particularly within a mouse with a functioning immune system, which again is quite complex and difficult to achieve. The thing that will help us is that Daniel Murphy, who's on this slide, has over the last 10 years developed, you wouldn't like me to say perfected, a genetically engineered mouse model that has got an intact immune system, that develops mesothelioma in a way that we would recognise compared to the human disease, and it will really function in work package 4 as the test bed for new drug target combinations that emerge, that we can actually test them in that mouse before they go into patients. And then the final work package is split into 2 parts, and I mentioned risk at the start. So the risk element is the top little cartoon there with the orange boxes. And so it will be tied to the meso-origin, to the prospective tissue collection programme. And what we will be doing there is in all the patients who have benign pleurisy who we recruit, or a proportion depending on the biomarker, we'll collect biomarkers in blood and in exhale breath and in perfusion MRI, and then follow the patients up. And we hope to be able to conclude with a signature that actually identifies the really highest risk groups that you might use, for example, as eligibility criteria for a trial of a new drug that emerges from the back end of the pipeline. And then the last bit is the little cartoon at the bottom with the pink boxes, is we have a whole work package focused around artificial intelligence, and particularly its use in imaging. The images, could we just blow that slide up just a little bit, just to show so still not huge, but hopefully you can see the panel at the bottom shows essentially CT scans with green rings, and the rings are either axial or coronal views of mesothelioma, where the colours show the tumour that has been outlined. And that process is incredibly laborious for a human to do. But we've, over the last 2 or 3 years, worked with Canon to develop an artificial intelligence algorithm that we've trained to do that, so the green images you see there have been drawn by an AI system that has never seen that tumour before. That you can't see it, but the image to the left is actually in red, is the human drawing that was used to train it. And so we're going to, within Work Package 5, we'll pull together 1,000 CT scans from across the UK using the NCME network. So we're leveraging all their infrastructure. NCME is the National Centre for Intelligent Medical Imaging, and Fergus Gleeson runs that. And so that will allow us to hopefully generate an AI tool that could be used not just, it really is for clinical deployment or for making clinical trials cheaper, because you don't need a radiologist to report your response. Oh, there you go, fancy. Yeah, there you go. So that was it, really. I'm going to kind of wrap up. I mean, some of these comments are not just about the accelerator, but are about, the clinical network. I think we have made a lot of progress. We've heard some challenging things, but we've also heard some really positive things. I think Dean's talk is incredibly exciting with what's on the horizon next year. And a big thank you to all those involved, including our audit staff, our QPI staff, our admin, and all the clinicians. the challenge really for Predict Meso and for research, I think more broadly, is better preclinical science and getting better drugs into trials. And you know, the title of Paul Krause's book is really what we're trying to get to, which is surviving mesothelioma. And I think this year, I think we're probably a little bit closer than we were last year for various reasons, even though the pandemic has been a bit miserable. And I'm going to stop there, and I'll